So let me give you a brief introduction to cognitive and affective neuroscience in the next few slides. First, let's talk about cognitive neuroscience. The goal of cognitive neuroscience is to understand exactly these cognitive processes that we talked about. So control processes and automatic processes that are in the cognitive domain. And that basically means those are things that are typically considered to be non-emotional. Cognitions like attention, memory, perception, and executive function have been studied quite extensively by cognitive neuroscience. And the question here is, what are the neural correlates of these cognitive processes? And so in experiments in cognitive neuroscience, such as in this one, you manipulate how much, let's say when we talk about attention, how much attention is paid to a stimulus. So you have one condition where the participant pays attention versus another condition where the participant doesn't pay attention. And by attention, cognitive neuroscientists typically mean the process of focusing on an item in your environment. This can be a visual focus, but you can also pay attention to a voice that you're hearing over some noise that is happening in the background. And you can pay attention to basically all of your senses or to specific aspects within all of your senses. But obviously the most commonly studied one is in the visual domain, such as in this task that's shown here. So in this task, to just briefly tell you, but I'll illustrate this in a second, you have dots that move in random directions, but a certain percentage of these dots actually move in the same direction. So let's say this is a downward motion and 50% of the dots move down, whereas the other 50% move absolutely randomly. And then the experimenter may ask you to pay attention to the direction of motion, let's say downwards in this case, but it's rightwards in the illustration here. And what's typically observed is that when the subject pays attention compared to when the subject does not pay attention and just passively views, you see this, well, it's actually called the attention network nowadays, consisting of frontal eye fields, inferior parietal sulcus, then some visual areas, as well as in this case, MT, which is a visual area that encodes motion. So it makes a perfect sense in this case that this network is highly activated under conditions of attention compared to conditions of where the subject doesn't pay attention. But let me briefly illustrate the task to you. So this is an illustration of the random dot motion task where you can see dots moving in a certain direction. They're a bit small now, but you can still see it as speckles. And you can see that the direction of motion is changing. You can also manipulate the coherence, so it gets a little bit harder to identify the direction of motion. But as an illustration, this is pretty good here. Now let's talk about memory. Now that we've seen attention, and there are two aspects of memory that I want to talk about. One is place memory, and the other one is amnesia, where we have observed memory dysfunctions. It's one of the disorders of memory that we know about. The first thing here to note is that the region across both of these different levels of research or different lines of research in the brain that represents memory is the hippocampus. And that's what I'm showing here. So this is a part of the hippocampus within the brain that contains these hippocampal place cells. And I'll show you a demonstration of this next, but basically what can be demonstrated is that within this region of the hippocampus, we have different cells that specifically encode a region within the space that an animal or a human is moving along. So, so these cells are color labeled, and they will be in the example next. So for instance, the yellow cell here fires when the rat is approaching this curve in this maze. So this is what's a typical maze. And the animal is asked to move around this to the end where he might receive a reward. And different neurons encode different areas of this maze. So the first turn is identified by these yellow place cells. Later on, it's identified by these red place cells. The second turn, let's say, and the third turn by these orange plate cells, where the color indicates that a different neuron is firing and encoding this area, this region in space. Again, I'll show you the illustration next, but let me quickly mention that there's a replication in humans of this, and what these researchers did is McGuire et al. Published this in Hippocampus. Well, the title of the journal is actually quite fitting in this case. They looked at taxi drivers within London, and it actually takes quite a lot to pass the test to become a taxi driver in London. You have to know the map of London quite well. And then what they did is they looked at the number of years that people had been driving a cab or a taxi within London and correlated this with the volume within the posterior hippocampus. So this area here. This is one of the, it's also an illustration of the wide ranges of applications of magnetic resonance imaging. So you can look at functional images that show changes in blood flow across time, but you can also look at the density of neurons from anatomical images that you acquire within the scanner, and that's what they did here. So what they show on the x-axis is the density of the neurons within this region, within this very region here, and on the y-axis you have the years of experience taxi driving, and they show that there's a positive correlation between this. So indicating that a taxi driver that had been driving for more years has a greater density within this part of the hippocampus that is responsible for encoding place. So indicating that potentially this is a reflection of neuroplasticity. So as you get more training in something, particularly something that is represented within this region of the brain, then the number of neurons, the neuronal density, increases in this region. So let me next show you the illustration from a rat walking through a maze like this, and how different cells start firing. So first off, notice that the popping noises, I didn't mute them because they're actually quite interesting, because they reflect an action potential fired by the neurons. So this is showing the maze in the middle. The rat is in the lower right-hand corner. And the important thing here is that you can see the rat walking through this maze. And once I stop the voice narration, you can also hear the cells firing. The color coding is such that each color represents a different cell within the hippocampus region. And you can notice that while the rat is walking through this maze, different cells start firing at different locations. So each cell seems to be associated specifically with a given region within the maze, helping the rat then navigate through the maze. Although there is some noise and some overlap across cells. But now let me allow you to listen to the firing. So now let's talk about another evidence for the role of the hippocampus in memory. So again, the goal of cognitive neuroscience is to associate specific functions, neural functions, with specific behaviors. And that's exactly what we're seeing in this case. So what happens here is that there's some damage to a specific region within the brain, namely the hippocampus. And then we observe a dysfunction in behavior in the form of amnesia. That means that patients with amnesia have difficulty forming new memories or remembering old things. So in the case of retrograde amnesia, these patients have difficulty accessing past episodic memories. Episodic memories are the types of autobiographical memories that are very important for us, for our personality, and generally to remember past things that have happened in our life. And anterograde amnesia is the inability to form new long-term memories. So once you have anterograde amnesia, you can no longer make new memories. And once you have, or a patient with retrograde amnesia, cannot remember things that happened in their past. So I will show you an illustration of a patient with amnesia in a second, to show how these symptoms look like and to show clearly that there's a relationship between the function of the brain and the abilities of the person. And it turns out that the hippocampus is actually involved in both types of amnesia. And the example is now from the patient that I'm showing you who has damage to the hippocampus and surrounding tissue. So let's have a look at this video. I apologize for the quality. It's an older video from a BBC program that was shown. This is a British patient, but it's an excellent demonstration of the effects of amnesia on the patient's life. So let's have a quick look.
One man is consigned to live entirely within the present, with terrible consequences. Clive Waring has the worst case of amnesia ever known. 20 years ago, he lost his memory, and now his wife, Deborah, is the only person he recognizes.
Clive really only has less than 30 seconds memory, and sometimes it's as little as perhaps 7 seconds. It's as little as a sentence.
I'm going to see your sister, Adele. Her daughter's got married recently in New Zealand. And so they're having a party.
Funny how the ladies acquire different titles when they get married.
Do you know who I'm going to see tomorrow?
Buckingham Palace.
No, really guess.
I don't know.
You don't know? Adele.
Oh, I see.
Do you know why I'm going?
No.
She's having a party at her house tomorrow.
It's her birthday, isn't it?
No.
Yeah.
Do you know why?
No.
It's to do with her daughter.
I see.
Do you know why her daughter's having a party?
No.
Guess.
No, I don't.
She's just got married.
Oh, I see.
She just got married in... Do you know what country she just got married in?
No, I don't.
In New Zealand.
Oh, I see.
Yeah.
The sentence he is in, he will probably have forgotten the sentence before. You ask him a question and he'll give you an answer, but while he's giving you the answer, he's already forgotten the question. That's how short it is.
I'm going to see your kids tomorrow.
You see my kids?
Yeah, your children.
What are they up to now?
Do you know what they're up to now?
No.
Guess what you think they're up to.
No idea, I couldn't guess. I don't know what their O-levels were. They hadn't got an O-level last time I was conscious. So I didn't know what possible thing they could do, did I?
No.
No, I couldn't possibly guess.
Where do you think they are?
No idea. I don't know where I am.
Yep.
All he has is void behind him.
It's been like death. I've never seen a human being before, never had a dream or a thought. The brain has been totally inactive, day and night the same. No thoughts at all. As far as I'm concerned, the doctors have been totally incompetent. I've never seen a doctor the whole time. Oh, look, it's come. Oh, you're wonderful.
That was a very musical kiss. I'm dizzy. I don't know which part of the room I'm standing in.
Can we dance?
Next, let's talk about executive functions. Those are typically defined as higher order cognitive functions, and they include things like planning relatively complex behaviors and looking forward into the future, as well as inhibiting prepotent behaviors, so behaviors that we would rather do, but in a given context are not allowed to do. So there are many social contexts that prohibit us from doing certain things, right? Which we can do when we're alone. So it's sort of an executive function that's a little bit more difficult. So these are controlling behaviors that depend on context and change based on the context, and that is the kind of cognition that is studied typically with the Wisconsin card sorting task, which I'm outlining here, but I have a demo for you guys in just a second. But let me show this to you. So this is an illustration of the Wisconsin card sorting task. You have a deck with 3 different symbols on it that differ in the color, the shape of the symbol, and the number of symbols. And then you draw a new card, and you're supposed to match it to one of these symbols of the original card deck or set of cards. And if I ask you to match this, you can give actually 3 correct answers if you don't know what the rules of the game are. You could say that, look, there are 2 items on the card, so it actually matches this card, where there are also 2 items on the card. You could also say, well, those are crosses, so it matches this card, which also contains crosses. And finally, you could say, well, they're blue symbols, so it matches the last card because it also has blue symbols. And all of these are correct, but at different time points in the task. And these contingencies change throughout the task. And you have to learn via feedback whether what your response currently is the correct one. And then it might be the correct one for a few moments. So let's say 2 is the correct answer right now. So you say 2, then you draw the next card, and it's 3 other symbols. So you say 3, and you say, yeah, that's you get the feedback that's correct. But then it changes. All of a sudden, numbers are no longer the correct response. And then you have to find the new rule. And this is a kind of task that engages a cognitive system. This is from a meta-analysis. Particularly, the lateral prefrontal cortex is involved in basically inhibiting this prepotent response and switching to a new rule and learning about a new rule. So the lateral prefrontal lobe is the region that can do these types of executive functions. But let me show you a demo of this task really briefly to allow you to see this task in action, basically. So this is from a website, scitoolkit.org, where you can try this out for yourself if you want. The link is actually in the slides. Here you see the different cards, and I'm trying out some stuff here to try to match them. And the question is it by color? Is it by shape? So it seems to be that the shape is the right answer right now. So I go with shape for a few times until this contingency switches again. At which point I get a little bit confused, but then eventually figure this out, as you'll see. I think the best thing to do is actually to try this out for yourself, because it's actually quite fun to do, at least for a minute or 2. You don't need to do this forever. And you can also skip watching this demonstration at any time that it doesn't interest you anymore. I think it's quite clear what happens here. So now the contingency switched. It's not color, it's the number of items on the card. And I get this right a couple of times. And I think at some point I get confused again. There we go. I got confused there. So you can see the task is not too easy. You have to pay attention to it continuously. Otherwise, you start messing up like I do here for a few trials. But then eventually I figure it out again until the contingencies change again, at which time point I have to relearn this then. Okay. So, given that cognitive neuroscience looks at all these different elements, the step to investigating higher order cognitive processes, from looking at attention, memory, perception, and executive function, was actually relatively small. And that's what I'm trying to illustrate here. So we had these different elements that we've already looked at. We had perceptual elements. We had motor planning elements that are involved in, for instance, the Wisconsin card sorting task. And the step from there to going to a more full-scale model of how decision-making might play out, which involves all of these elements. It involves attention, it involves perception, it involves motor planning, and it involves evaluation of the choice options, is actually relatively small. And I'm showing you a model here by Antonio Rangel that was published in Nature Reviews Neuroscience in 2008 that reflects this. So the model basically argues that at the bottom of at the first level of a decision is the representation, which is usually a visual element most of the time in fMRI tasks anyway, but you can also be given a choice verbally, for instance, right? So then it would be an auditory element here. And that's basically trying to figure out what is the set of feasible actions right now, what are my internal states, what do I want. Next, you evaluate the choice options, which happens here in these regions, ventral striatum and VMPFC, but medial. So this is a lateral view. You would see this medially otherwise. Once you've evaluated your choice options, you're selecting one action that will lead, or maybe a series of actions that will lead to the desired outcome. This is part of motor cortex here. Then you get the outcome from your decision. You've done your action to obtain it, and in the scanner that's just pushing a button. In the real world, it's most of the time something more effortful, like walking some distance to a more favored relative to a less favored restaurant to get a better meal. Then you receive the outcome of your decision, and you might like it more or less than you expected. This is actually called the reward prediction error, which we'll talk about in one of the future lectures. And the difference between what you expected and what you actually got then becomes part of learning, which can be used to update your predictions of how you value things for the future. So this is a relatively simple model of the steps that at minimum should be part of a decision process. So this is the part on cognitive neuroscience that I will finish here for this video lecture. Next, we will talk about effective neuroscience.